time. Okay. All right. So there's three of you?
Yeah. For now, yes.
Okay.
Maybe
Okay.
someone will arrive later.
Okay. All right. So when last time we were talking, we talked about these inherited tumor suppressor defects, and the where we left off was right here, BRCA genes, which are defective in breast cancer. That's what the name stands for. P16, which describes its size. Most things that begin with P and have a number after them, that's the molecular mass in thousands of Daltons.
Okay.
So P16 means that protein is 16,000 Daltons. Okay? So P16, which is a defect that's found in a lot of melanomas, and then the adenomatous polyposis coli, or APC gene, which is mutated in this familial form of colon cancer, where the people get thousands of teeny little polyps all over. Here you can see some bigger ones on the top of the screen that are hanging off of the mucosa. So what happens is these things form on the surface of the colon, and what happens in the colon is that the feces, right, in the colon, if you think about what happens, the function of the colon is to resorb water, right? At that point, the nutrients are already out of the fecal stream, and what your body is doing is recapturing the liquid, right? And so you have a pretty hard substance that's flowing past over and over and over again with peristalsis. This stuff is moving past this bump, and eventually it pulls. And can you see where my cursor is? You see this like little mushroom looking thing, right? It's got like a mushroom, almost like a cap, and then like a little stalk. See that?
Yes.
Okay. And that's what polyps look like. So a polyp is something that protrudes into an opening. So you can have a polyp in your nose. It doesn't have to be in your colon. Okay? And a polyp is a general term. And what happens is the feces, the poop, essentially, right? As it goes by, it pushes on this, and it pulls it away from that flat lining. And that's how you get those stems like that. And when people get larger polyps, if you get single polyps, not this inherited form of cancer, but in people that develop polyps when they're older, a lot of times they're a lot bigger. And the way what happens is eventually the feces will tear that thing. It actually makes them bleed. And that's why people get blood in their stool. And that's one of the major ways that the colon cancer is detected. Right, is because they bleed. But that's why they bleed, right? Because these things are being battered in what's called the fecal stream, right? The poop, that semi-hard stuff is getting pushed by. Okay, so the next thing that we're going to talk about is essentially the superhero of tumor suppressors, right? So this is a cover of Science, and in particular, I'm going to show you a new article that just came out, but can you see the date for this one?
It's 1993.
Yes. Okay. So 1993. Okay, so a long time ago, P53 made the cover, right? It was actually named the Molecule of the Year. There's only so many molecules that can claim that, right? So you can only imagine how proud P53's parents were.
Very, very proud.
So P53 was on the cover for a reason, and they say here a genetic key to cancer. And at the time, right, if you remember when we started talking about when these tumor suppressors and oncogenes were discovered, right, that was around 1980. Remember the dates, 79, 80, 81, right?
Right.
So at this point, they had been working on tumor suppressors for probably 8 years, 10 years, something like that, because remember, they came second. And they discovered this one that seemed to be defective in so many cancers. It seemed to be a real key, and they thought, wow, we're really close to fixing this cancer thing. Okay? And so it was discovered initially in 1979. And so P53 was found as a protein that was complexed with, i.e. bound to, stuck to, large T antigen in cells that had been transformed by simian virus 40. And I mentioned that briefly when I talked about polyomaviruses. I said it doesn't normally cause cancer in humans, but it can transform cells. And these SV40 transformed cells were used to grow a lot of things, make cells grow, even animals, not necessarily human cells. But they found this protein that was stuck to this protein, large T antigen from SV40. And we'll talk about why they were stuck together in a little bit. But that's how it was first found. And then when they... He wants to... Come on. Go out. He's running out the door now to go chase that. Yeah. Okay. And they found that this protein, P53, was found at very high levels in transformed cells. So here are the malignant nuclei. This is a cancer cells from a patient, and they stained it for P53, and you see that the nuclei of these cells just turns black, right? So there was a ton of this P53 protein in there. And what would that suggest to you? Based on what we have talked about in class, would you expect to, if P53, if there's more of it in cancer cells, what kind of protein would you think it was?
An oncogene.
Yeah, right? You would immediately say, man, this thing is really elevated. There's much more of it in cancer cells. So this is exactly what they thought, right? They saw this stuff as elevated in these cancer cells. They said this thing is an oncogene. So they did gene transfer experiments, okay? And when they did that, when they took, just like they did with RAS, like we did with these other ones, when they took the gene and they put it into cells, they showed that P53 was in fact an oncogene, right? So this thing acted like an oncogene. It transformed, was able to transform cells. But I'm telling you, and you're just going to have to believe me on this, that this P53 is in fact a tumor suppressor. So what could be wrong if you do a gene transfer and you put it in and you say, yes, it's an oncogene? What could be wrong with the experiment? Why would this experiment maybe not work?
Because it's supposed to grow instead of the other way.
You're going to just have to repeat what she said, Ayana.
She's saying that it's supposed to grow instead of like triggers that growth and instead of the other way. Or...
Yeah, well, I mean, when they did the experiments, it looked like an oncogene. It made the cells look, you know, abnormal, right? Transform. But we know there's a problem with the experiment because we now know, looking back, of course, that P53 is a tumor suppressor.
But it was the experiment that went wrong?
It wasn't. They did the experiment right, but why could the results be misleading? When we transferred RAS, what were we putting in? What kind of RAS? Where'd they get it from? Or SARC?
RAS?
Right? So the gene SARC that we transferred, or RAS, they got it from cancer cells, right? It was from that EJ bladder cancer, remember?
Oh, yes.
So these are mutant, and it turns out that the P53 that they first purified was in fact a mutant form.
So the original was supposed to suppress, but the mutant
Yes.
made a different
Yes.
function.
Yeah. They accidentally, without knowing it, when they purified the form of P53 that they found, they were working with a mutant version. And it turns out that and I'll show you in a minute. I'll tell you briefly, and then I'll show you a picture in a minute. But P53, when it works, the protein doesn't work by itself. It works as 4 copies of P53 together, right? So the term for that, the specific term is that it's a tetramer. Tetra means 4.
So it's made of 4 P53 versions?
4 copies of the same P53 protein, 4 units, right? So it works as an oligomer, right? It is a protein that works, as a group. And it turns out that the form that they worked with, this mutant form, causes, if it combines with normal copies, it actually makes them not work, right? So if you have one bad copy in there, the whole thing doesn't work. And a mutation that causes a normal copy to not work has a specific name. It is something you should probably remember. You will see it again in the field or in biology in general. It's called the dominant negative mutation. Dominant negative. In other words, it's inactivating the protein, but not only does it inactivate that copy, it actually can interfere with the activity of normal copies. You got that?
Yes.
Yeah.
Okay, and that's called a dominant negative mutation. They do exist, and they're very interesting, and in particular with this one, that's what they were working with. So if we go forward, and I'll show you kind of what it looks like here. In '89, Vogelstein did a survey of lots of cancers, and he found that P53 was mutated in lots of cancers. This has been substantiated over and over again. It turns out that P53 is defective in about 50% of all cancers of any origin. It's one of the highest for any gene, right? 50% of all cancers have problems with this gene. It's thought that all cancers have a defect in the pathway that P53 controls. Right? So even if P53 itself is not defective, there is something downstream or upstream that's broken. Okay.
So that's the most recent hypothesis of what drives cancer.
I mean, this is the most commonly mutated tumor suppressor. I mean, what drives everything is genetic instability.
Right.
And accumulation of mutations and selection for new phenotypes.
Okay.
Right?
There's like a factor that makes P53 to get mutated.
I mean, smoking does that. Environmental mutagens, random mutation. It just seems to be that this protein, and we'll talk about it in more detail, controls so many different things that when this particular protein goes bad, the cell has a very hard time recovering. So we looked at a subway map, right, of cancer pathways, but you really have to think of it like a huge mass transit line with all kinds of bus routes and taxis and everything. So in most ways, if you were going home and there was a bad, if there was a traffic jam or an accident, right, you could probably say, oh, well, I'll turn left here and turn right here and I'll still get home, right? It may not be the best way, maybe not the most efficient way, but you get home. What I'm saying is that it seems to be with P53 that when that pathway is broken, there really aren't any alternatives. And so the cell is in real trouble.
Okay.
Okay. All right. So P53 functions as a tetramer, right? Okay. And this is what it looks like bound to DNA. Here's a copy of P53. These are some of the more common mutations, the ones that are in red, which are highlighted by arrows. And if you'll see, they're the ones that actually interact with the DNA, right? Because this is our helix, right? This is our double helix. So P53's function is a transcription factor. It binds to DNA and it turns on or off the activity of target genes. There is a specific binding sequence that it recognizes. It reaches into the helix, into the groove. It interacts, and it causes transcription. Okay, that's what P53 does for a living.
Okay.
Now, there are lots and lots of alternative forms of p53. And so the top one, we're not going to talk about all of these. I'm going to show you one that was just discovered in the end of July. It was just published. Okay. So they've been working on this since 79, and we just discovered something. Okay. So here's wild type p53. This is what it looks like. It's got DNA binding regions. It's got different forms. There are a bunch of different ones, sometimes using different promoters, sometimes alternative splicing. Are you familiar with that? You don't always take out the same introns when you process an RNA. Mm-hmm. So that's called alternative or alternate splicing. Okay. And so there's multiple different p53s. And this is one that just came in, again, that I just mentioned. This was just published online, so it's not even really in the journal yet, I don't think, in the end of July. And this is a form of p53 called p53 psi, right? The Greek psi.
Right.
And this is transcriptionally inactive, so it does not target. It doesn't work to target the promoters, but in another way, probably because it's able to bind with other copies and able to interact with other proteins, it actually programs cells towards metastasis, right? It makes them more metastatic. It actually drives metastasis. So this is a brand new observation, and it kind of goes against, of course, the grain of p53 as the good guy, of course. But we'll learn more. I mean, this just came out. Okay. Okay. So what is p53 doing? I like to use the analogy of a spider on a web. Okay. So here is a spider on a web, right? If you think of a cell so what does a spider do? This spider is sitting in the middle, and it's got its legs on these different, you can even see how this thread is bent down. You see the leg is pulling this down. I don't know if you can see the thread on your screen or not, but it's pulling it down, right? And it's kind of using that to keep this tense. The spider is sensing changes that are out there, right? If there's something that happens in this area of the web up here, the spider's going to know because it's going to transmit, right, down there and to the leg. The same thing is what it's doing with these other places. It's sensing all different things. So p53 acts the same way, okay? So picture our p53 here in the middle. Here's our spider, right?
Right.
And these are the things that p53 is sensing one way or another. And I'll show you some ways it happens, not in great detail, but just so you understand why it's so critical. p53 controls several decisions, right? It controls whether or not cells divide, right? Is this cell going to make two cells, right? Is the cell cycle going to progress? So it either arrests the cell or allows cells to proliferate. It is able to affect DNA repair. So if there is damage to chromosomes, or if the chromosomes are not aligned properly in mitosis, that signal will be sent to p53, and p53 will try to recruit proteins and affect repair. If repair can't happen, right, then the cell is going to be triggered to kill itself. And that's apoptosis. Right?
Right.
So you're either going to block the cell, stop it from doing anything, or you're going to kill it.
Okay.
Okay? And p53 is also involved in inhibition of angiogenesis, something we'll talk about a little in a few weeks. Okay. Now, how does it make those decisions? p53 interacts with a lot of proteins. That's why it's so hard to deal with it when it's gone.
Right.
Right? It senses whether or not there are enough nucleotides around, because if there aren't nucleotides, you're not going to be able to continue synthesis. There's no reason you're not going to be able to divide a cell. It gets information as to whether or not UV radiation is present, whether or not there's radiation damage to the DNA, ionizing radiation, which causes a different kind of damage, whether or not oncogenes, and this is critical to what we're talking about for most parts, and I'll show you a diagram in a moment, whether or not these oncogenes are sending signals when they should not be, right?
Right.
Whether there is a lack of oxygen, right? If you don't have oxygen, you don't have energy production, and you're in trouble.
Right.
And whether or not there's a blockage in transcription. So if you can't transcribe proteins because there is some damage to the replication transcription machinery, if the DNA is damaged, then you're not going to be able to transcribe. Right? I mean, how does the RNA polymerase go by something if you have DNA damage? It doesn't know how to transcribe. So DNA damage blocks also transcription. And so if that happens, then P53 is going to put the brakes on this whole thing. Right? So it seems to be a critical integrator of a bunch of different signals. Now, we'll talk about it in a minute. There is no great explanation from an evolutionary standpoint, or like if you're going to say, why is P53 the one that does it, or how come so many things go through this one protein. You know, you're going to have to talk with a couple billion years of evolution. I can't answer you. I don't know the answer. No one knows the answer. But it seems that, and I'll give you a possibility in a few minutes, but P53 seems to be a critical juncture that integrates signals from all over the cell.
So it really is a very particular protein. Like there are no substitutes for what it does.
No, there are those alternatively spliced forms, and then there are a couple related proteins called P63 and P73. Obviously, they're just a little bigger, right? Related proteins, but they don't seem to be as important. There doesn't seem to be a good substitute or workaround.
Okay.
Yeah. Yeah, if I was designing the system, I mean, if you were doing it, right, if it was intelligently designed, to use a creationist term, right, if a cell was intelligently designed, you would have redundancy, right? You would have it so that there are several different things that could do the same thing if it's so important. But that's just not how it works.
Yeah, unless you think that it's never going to fail, then you don't need
Right.
substitutes.
So, okay. So what does P53 do? Transcriptional activation, I said it binds to promoters, it binds specific sequence and turns on transcription. It is able to shut down transcription of some target genes. And it does have transcription independent activities. And so when you see, I'll show you in a moment, it can interact with a lot of different things. It's not surprising that P53 can have activities that really influence the cell that don't involve binding DNA. Okay, it's a multifunctional protein. So transcriptional regulation is just one of the things that it does. And so it can both drive apoptosis, or it can sort of block apoptosis if you want to kind of pause and see if you can fix things, right? P53 will do that. And it seems to have roles outside of the nucleus. These are much less well understood in apoptosis and in autophagy. And autophagy is something that they don't typically cover in a lot of introductory courses. Autophagy means, auto means what?
Cell.
And phagy, do you know what that is? Like macrophage. Do you know what phage?
That you eat.
To eat. Yeah, right? Hyperphagy means to eat too much, right? Okay. So autophagy means to eat yourself. And literally what they're talking about is the digestion in the cell of defective old proteins in organelles. Like, for instance, mitochondria are subject to lots of damage. They turn over, and rather than start from scratch, your cell will envelop them into little vesicles and break that down and reuse the parts. It makes sense, right? It's efficient. You already obviously had everything you needed to make a mitochondrion, so why start over again? So autophagy is a way that cells recycle things inside. Okay? And P53 seems to be involved in that somehow. Again, this number 5 is something that people are finding out more about now. It's not really clear as well. Okay. Now, so if you have low normal amounts of P53, you're going to get these are the different triggers you could have for P53. Here are the responses, and essentially, if things are okay, right? You have all the nutrients you need, you got all the nucleotides. It's like a checkoff list, right? Is everything going okay? Then essentially, you're gonna, maintain stem cell. You're gonna have cell reproduction, stem cell maintenance. Everything's fine. If you have low levels of stress or damage in the cell, then P53 is going to essentially put the brakes on. You're going to undergo repair. You're going to replace those damaged organelles via autophagy, right? That seems to be the link there. It's going to help with relieving stress in the cell. And by stress, what I mean is anything that causes the cell to behave abnormally. That could be reactive oxygen species, oxygen radicals, radiation, chemicals, anything.
Right.
Okay. Not like traffic jam. That's a different kind of stress. Yeah. Right. And so if you have low levels or moderate levels, then P53 essentially says, wait a minute, let's see if we can fix this. It makes sense. Right? And if you have severe stress or you have a lot of oncogenic signaling, and we'll talk about that in more detail later, but if you have a lot of oncogenic signaling, then you're going to undergo senescence or apoptosis, and this is different ways that cell can die. There's apoptosis, and then there is necrosis. And necrosis is typically thought of as being different than apoptosis, this programmed death, but now people are seeing a lot of connections. I mean, normally when you think of necrotic, you think of like, you slam your finger in a car door and it rots and falls off or something. That's, it's black and yucky, right? That's necrosis. That's because you damaged something and the cells died. Apoptosis is typically thought of as a cell being one individual cell or a group of cells being told to die, and they do in a very neat time, manner, right? But now we're learning that there's some overlap. Okay, so P53 is as you would imagine very tightly regulated, right? If you have a protein that's this important, you're going to regulate it pretty strictly, right? And so this is some of the regulation. So let's start here in the middle. Okay? You got it?
Yes.
I'm not sure how clear your image is. So, okay. So here's P53. If we have damage, right? If there is DNA damage, okay, then you activate proteins, you transcribe or activate, post-translationally activate proteins that will modify P53, typically by phosphorylation, but there are others, but they'll modify P53 to make it active, and it can then go and turn on or off a bunch of target genes. Okay? Got it?
Yeah.
Okay. But you obviously need to have this thing turned off when you want it turned off. And P53, because it's so crucial, it's really tightly regulated levels. And so one of the things that P53 regulates, one of the target genes, is a protein called MDM2, a gene, obviously, that makes a protein called MDM2. And MDM2 actually gets its name because, remember we talked about gene amplification, making too many copies?
Yes.
That's how MDM2 got its name, because it was found at really high levels in copies of the gene in cancer. and it formed what's called a double minute, which is a little teeny tiny little chromosome. It's not a real chromosome, it just contains all that amplified DNA. And they call those double minutes. And so this gene is called mouse double minute, MDM. And so it was found in cancer. And so MDM2's job is to bind, if we follow it down here, MDM's 2 job is to bind to P53, and it drags P53 over to a proteasome, gets it ubiquitinated, polyubiquitinated, and feeds it into a proteasome where it gets destroyed. So are you familiar with proteasomes?
Some.
No?
No.
Okay. So I'll tell you briefly. The organelle, it gets mitochondria get all the attention for some reason. I don't know. So here's what a proteasome looks like. You see down here with the yellow?
Yes.
Okay. So it looks like a cylinder, like a Coke can shape. And then on the ends, on both ends, these would be the same. On both ends, this thing is kind of symmetrical. It's got 2 ends and it's got this middle. In the middle, it's got a bunch of proteases, a bunch of proteolytic enzymes. And you know what an incinerator, a garbage disposal in a sink?
Oh, yes.
Right? You put the stuff in, you throw the switch and it chops it all up. That's what these things do to proteins. So what happens is the protein gets brought over, it gets a bunch of ubiquitin. Ubiquitin is a protein, a small protein, about 60 amino acids. And you get a chain of those ubiquitin molecules added to the target protein. And then the ubiquitin comes over here, and this protein, or in this case, a bunch of them, is fed into this disposal unit. And it gets chopped up into little pieces. So this proteasome, the proteasome is a target for cancer treatments. Bortezomib is a drug that blocks this in cancer. Okay? An approved cancer drug. So protein degradation, in class, when you take intro bio, you always stop at transcription, translation. You never actually get rid of anything. But here, in real life, we have to get rid of proteins. One of the ways is through autophagy. Another of them is through this proteasome-mediated thing. Are we good?
Yes.
Okay. All right. So MDM2 causes P53 to be destroyed. And as you see, P53 causes MDM2 to be made. So when P53 is first made, one of the first things it does is it turns on the gene that's going to eventually turn off its activity. So it's a negative feedback, right? It limits itself.
Right.
Okay?
Okay.
Okay, good. Now, P53 has friends too. Okay? If you're that important, you have friends in high places, right? So this is P53 we're talking about, right? So P53 has a protein that works to help it called ARF. And all of these are on your chart that I gave you. I know you can't have all this stuff up at the same time, but trust me, it's there. Okay? So MDM2 and ARF are on your diagram that you have. And ARF is P19, so we know what that means, right? A small protein, P19, ARF. ARF stands for, and I tell you this not for you to memorize it, but just because I feel like if I tell you, it'll maybe make some more sense. So you can forget it, but I'll tell you anyway. So ARF and another protein come from the same exact gene. The same transcript, essentially, is read in an alternative reading frame in the gene. Mm-hmm. And so ARF stands for alternative reading frame. Okay. That's what the protein is named after. There's 2 different, and it just so happens that both of the proteins that come out of that gene are both tumor suppressors. Okay? So what does ARF do? ARF binds to MDM2. and it actually takes it over to the nucleolus, right? Those dark areas inside the nucleus where the ribosomes are synthesized, and it sequesters MDM2. So if it does that, MDM2 can't bind to P53, so P53 doesn't get degraded. So ARF, it turns out, we now know moving forward, ARF is one of the key responders to oncogenic signaling. Because if ARF signaling goes up, that means it indirectly makes P53 more active.
Right.
Okay?
Okay.
Okay. Now, P19 is the mouse version. P14 is the human version of the same gene, and there are a little bit of differences, but for our purposes, they're pretty close. Okay. So here is a circuit. Okay. So this puts all of the things that we talked about in place, mostly. We'll introduce a couple new ones. But here we have P53. If it turns on P21, which is one of the very first things, major target, in that Science article, that was the only thing they thought it did. They thought they knew P53. They put an arrow from P53 to P21. Done. Okay. Turned out to be a little more complicated. Okay. But P21 is a cyclin-dependent kinase inhibitor. So it blocks the cell cycle and drives cells into senescence. Okay? So this is one thing that P53 can do. Another thing that P53 could do is to turn on BH3 proteins on your diagram in your the map that I gave you. I think the one on there is BAX, B-A-X. I'm pretty sure is BAX. It could be BAC, but I think it's BAX. That's an example of a BH3 protein, and those proteins cause death by apoptosis. Okay? BAX or BH3 is a very generic term. BAX is a specific protein.
Okay.
So, but these BH3 proteins, most of them will drive cells towards death. And MDM2 is blocking P53, right? P53 is turning on MDM2. So here's our negative loop, right? And it seems to be that one of the critical regulators is ARF. And ARF integrates signals from oncogenes. Here's one that we've met, RAS. Okay, again, mutated in about 40% of cancers. So a really high percentage of cancers are dependent on this pathway being broken, overactive. Another oncogene example that turns on ARF is MYC, and MYC is transcription factor itself. So here is sort of a cycle. If you get these on, then you're going to, if RAS and MYC are on, you're going to be able to drive cells into S phase and get proliferation. If they're not able to block ARF and there's a lot of signaling, then P53 will lead to either senescence or apoptosis.
Okay.
Does that make sense?
Yes.
It is complicated, but you'll take some time. Right? I mean, it's not simple. Okay. Now, how does P53 do all this stuff? Well, it turns out that of all proteins, right, it probably interacts with almost more than any other protein. And it is one of the most heavily modified proteins, okay, in all of proteindom, right? So here are some examples. Here's P53. It has a transactivating region. That's the region that binds to other transcription factors and tether the polymerase. It's got the part that holds it to the DNA.
Right.
It's got negative regulatory regions, and then it's got other binding regions for other proteins. And here is some of the proteins. This CHEK1 and ATM and ATR, these are DNA repair enzymes. Here's where they phosphorylate. DNA, PK, another protein kinase phosphorylates here. MAP kinase does here. HIPK here. This is a translation activating factor. The Janus kinases. This thing gets phosphorylated all over the place, and that's in the first 100 amino acids. This is what it looks like if you look bigger. This is the one I'm going to put on your test. Okay? All right. So again, not to be memorized in any way, but you can see there are lots of sugar modifications, kinases, serine kinases, threonine kinases, all of these proteins. Look at all the proteins that interact. And here is the one, SV40T antigen. Here's the one, how they discovered it. Okay? Now, just to go backwards, remember we talked about viruses? What do viruses do when they go in cells?
They copy their genetic material.
And how do they do that? What do they have to do to the cell?
To the cell, like...
Yeah, I mean, they take over, right? I mean, you don't want the cell to kill itself. You don't want the cell to do cell stuff. You want to do virus stuff, right? And so it turns out that this SV40T antigen, for instance, the reason that they found it stuck to P53 when they discovered it was because it binds to P53 to shut it down. Right? So this T antigen, or tumor antigen, binds to P53 and prevents it from shutting down. It won't let the cell go into senescence or apoptosis. If you're a virus, that would be a terrible thing, right? So that's how that works, right? Here's BRCA1. I'm not going to go through all of them, right? But you see the ones that you know, why they're so important. So it's one of the most highly regulated and most interactive proteins known. Okay? Are there familial cancers with P53? The answer is that it is very rare. It's called Li-Fraumeni syndrome. 70% of them have mutations in P53 in an inherited manner. Many of them, lots of different types of cancers. There are very, very few of them. There's less than 500 families in the whole world that are known with this disease. And they get cancer very young in typical, right? So half of them before they're 40. You don't tend to see inherited diseases in tumor suppressors too much if they're that important, right? If it's that critical, most of the time that mutation is probably embryonic lethal, right? You never get to see a baby, right? Other familial cancers, we're going to walk through a couple of them. This is one that's been studied very well. It's called xeroderma pigmentosum, or XP is how it's referred to. It is a defect in the nucleotide excision repair DNA repair pathway, right? And in particular, the genes that are defective are the ones that excise the damage caused by UV light. And so these people are very, very prone to getting skin cancer. They're really extreme, I mean crazy sensitive to UV light. And they also have eye problems because of the damage. So this is what someone looks like with XP. This is a young boy. You already see that he has sort of precancerous or cancerous lesions on his face, okay? And that's because the ultraviolet light is causing pyrimidine dimers. If you remember, if you learned about the mutations, what causes them. So adjacent pyrimidine nucleotides are fused together essentially by that light. It causes pyrimidine dimers form. And these kids cannot repair that damage. And so this is actually, I thought I would show you, this is actually a thing called Camp Sundown that they have here in New York. And what they do is, this is for kids with XP, and they do everything at night. right? So all of their activities are in the evening. All the kids there have XP, so they all do stuff at night. They go out. All the lights inside here or in a school with a student with XP, they have to replace. Even the fluorescent light bulbs have to be changed to ones that are low UV emission. They're very, very sensitive. All right. Okay. Here's another one just to give you a couple. Wilms tumor is an inherited cancer of the kidney. It's a transcription factor. This is what the protein looks like. It's on chromosome 11, and here's what the cancer looks like. So almost all of these hereditary cancers, they tend to occur pretty early. So here is a list of them. Again, we don't have to go through them all, but there are a number of these inherited tumor suppressor defects, right? We don't really have any familial diseases of inherited cancers associated with activated oncogenes. Like there's no inherited cancer associated with an inherited activated RAS or SARC. Why do you think that is?
¿Por qué no se heredan? Not associated with oncogenes.
Why would you not be able to see a hereditary form? Right, we saw it. Here we have all the hereditary forms of cancer that are due to defects in tumor suppressors, but there aren't any associated with defective oncogenes. Why?
Well, maybe they wouldn't allow life to develop correctly. I don't know.
No. Say that again with great authority.
They wouldn't allow life to develop adequately.
You're dead. Exactly. You're dead. You're dead. You never make it out of the embryo stage, right? I mean, if you have RAS going all the time in an embryo, you're in serious trouble.
Okay.
Right? I don't even know if you get to look like a fish even, right? So these are lethal events. Right? Continuous signaling from an oncogene in development would be disastrous.
Right.
Okay, so you really, really don't see it. Okay, so what do other tumor suppressors do? So I showed you only one, or in detail, regulation of the cell cycle. We saw both with P53 and RB, right? That's what RB does. Their DNA integrity, they're making sure that the DNA is not damaged, and essentially as an extension of that, they are protecting genetic stability, right? Remember, because we know that instability is the driving force for the changes in cancer. So it's really important. So DNA integrity, right? The stability of the DNA. They're involved in protein degradation, because if you have something that's supposed to stop signaling, you have to get rid of it. In responses to hypoxia, which is a low level of oxygen, we'll talk about hypoxia some more a couple times. Right? In the control of cellular senescence, that is the essentially off state of a cell that is not dead, but it's not dividing, right? Or they can actually kill the cell. They're involved in cell adhesion, cell motility, and we find new ones all the time, right? We find new ones all the time. So here is, again, sort of the hottest of the research. If you guys were starting your own lab, you'd be writing for grants on this, right? Because this is hot stuff. Looking at microRNAs, right? These are these non-coding RNAs that we talked about before when we talked about oncogenes. They can either be from their own promoter, or they can come from within an intron and be spliced out of an intron. And so any of these tumor suppressor microRNAs, their job would very likely be to inhibit the transcription or translation of an oncogene. That, by definition, would make it a tumor suppressor. Okay, and there's lots of them. right? Methylation, silencing, etc., etc. Okay? I was just showing that it was multiple different ways.
Mm-hmm.
Nothing particularly amazing about these articles. It is just to show you the variety of things that these microRNAs are do. Okay. Okay. So, importantly, right, not all tumor suppressor mutations are fully recessive. And so you really have to have one of the things that you guys, not for talking with patients, but for your own understanding of biology, is to understand, don't memorize, but understand what dominant and recessive means. Okay? Because I think people memorize those terms and they don't really know what they mean, right? I mean, you look at it as a little a or a big A or something, you're not really understanding it. So if something is dominant, right? If you picture only 2 copies in our cells, we have 2 copies of any given gene, for most genes, right? We'll just make pretend. 2 copies, right, of the gene. If you change one of them, even if it is the removal of an activity, if it's the addition of an activity, right, like an activated RAS, or if you lose the activity of something, if you make one change to one of those, and you see the phenotype at the cell level or at the level of the individual, that's what dominant means. It doesn't mean it's doing anything aggressive. People tend to think of dominant like they do people. Like dominant means you're kicking the ass out of, something else, right? You're stomping all over that other gene, But in fact, the dominant gene can do nothing at all, right? If the surviving copy that's there, so now we have one of each. Got it? We have one good copy, we have one bad copy. Mm-hmm. If we look at that person and we say, what phenotype does that person have? If the one bad copy shows up, then it's dominant. If the good copy is what you see when you look at the person, then it is what is dominant. And this mutation is recessive. Recessive, to flip it around, to define it another way, recessive means you need to have 2 identical copies of that in order to see the phenotype. Mm-hmm. Are we good with all that?
Yes.
Okay, because I didn't teach you genetics, so I didn't get to beat that into you when you were learning genetics. Okay? Okay? But that makes sense, right? So RB, for instance, at the cellular level, when we looked at it, RB is essentially recessive, right? That cell acts in an abnormal way. Right? But the cancer appears dominant. Remember we looked at that pedigree because of that. But that mutation is recessive, right? What did I say? I hope I didn't say it wrong. The RB is recessive, right? So if you have a single copy, the cell is still okay, which means that the mutation is recessive. You don't see it unless they're both broken. Okay? Hope that came out right. Okay. All right. But not all tumor suppressor mutations are fully recessive. So sometimes these things, so in other words, one functioning copy of the gene is not enough to make up for a bad copy. That's just another way of looking at it, right? It's not able to pick up the slack, right, for the bad copy. And so some tumor suppressor mutations are not this says RR. Sorry, that's a typo. It should just say one of them. R, yeah. Transcription factor like SMAD4 or P27, which is another cyclin-dependent kinase inhibitor like P21, or P10, which is, I don't know that you remember, but we looked at P10 as that phosphatase when we looked at PI3K and P10 went the other way. Remember we talked about that balance?
Yes.
Right? When we were putting that extra phosphate on that PI3K? Okay. So not all of them are like that. Just so you understand. Sometimes one of them will have a phenotype. The term for that, right? The term for the fact that one is not enough is pretty self-explanatory, really. The term is haplodiversity. insufficient. In other words, when you're haploid, not diploid, a haplo means 1, diplo means 2. When you have just one copy of that tumor suppressor, it is insufficient to make up. Right? So if you take out one, you're going to get a phenotype. Very likely, if you take out the other one, the phenotype is going to get worse. Okay? But anyway, this is a decent term for you to understand. Okay. Okay. Now, you can lose function by epigenetic changes as well, right? You don't have to mutate. You don't have to have a deletion. You can have DNA methylation, histone modifications, acetylation or deacetylation. You can even have, histone phosphorylation. We know there's lots of things that happen that control the activity. So, and then there are other interactions. There's modifiers of tumor suppressor function that can influence them, microRNAs being one example, or the environment, either other cells or the extracellular matrix, dietary factors. There's lots of things that will can sort of tip that balance. Okay. All right, so one of the questions then, and I addressed this before, is why are cells so dependent on these individual genes? It seems like a terrible design, okay? If you were building a bridge, you would never want the whole bridge to be dependent on one support, okay? I mean, it's distributed load, right? I mean, that's what you'd want to do. You'd want to distribute the load. So here, let me move this thing out of the way. So here is part of our map. Here's APC right here. That's that gene that's defective in the colon cancer, right, with all the polyps. Okay. Here's MYC, one of those oncogenes I just introduced before, the transcription factor. Okay. All right. And this is a receptor here, frizzled. This is like a growth factor, and this is a signaling pathway that goes through APC. So APC is an example, like P53, of sort of an almost overdependence on particular proteins. So most colon cancer cases have defective APC. I believe, don't quote me, but I think it's like 80 to 85%. So a large percentage of colon cancer cases have a defect in APC. It is a massive protein. So the average protein, Joe protein, right? Joe average protein has 400 amino acids or so. Probably a little bit shorter than that. And this one has 2,800. Okay, so it's a very large protein, and it has, like P53, you see a theme. It interacts with a bunch of different things. It interacts with beta-catenin, which is a structural protein, also a transcription factor. It binds to microtubules because it's actually involved in sensing extracellular connections and movement and cell adhesion. Okay, so it binds to a bunch of different things, axin and just all kinds of things. Okay, all kinds of things. So it's a huge protein that interacts with a lot of different stuff, and it's right involved here. MYC is a very potent transcription factor. So if this APC isn't regulated, then you're not going to stop beta-catenin, and so this signal is going to be on all the time. You just put a brick on the accelerator. Okay? And so it doesn't make sense, right? This is how the signaling works. I'll just show it to you real quickly. Sorry. Here's the pathway in, I don't know where to put this. There is no place to put you. I'll put you in the middle. Okay. So here's what it looks like. In the absence of the Wnt, that receptor, the growth factor, I mean, frizzled is over here. These names, by the way, anytime you see a name like frizzled, you know you're talking about something that came from a fruit fly, because they're the only ones with any imagination. So the fruit fly mutations, what happens is you're breeding flies, right? Picture this, you're breeding flies. And you're looking at your flies through the microscope, and you get one whose hair is just all like, all the little things are just all frizzled up, right? And so what you say is, wow, that one sure looks frizzled. Okay? That is exactly how it happens. And so the genes are named almost always. Think about it though. I mean, it'll help you later. They're named based on the phenotype of the mutant form.
Okay.
Right? Normally the fly wouldn't have frizzled things, right? We discovered that gene because we saw what happens when it's broken. Right? The cystic fibrosis gene in humans. It's called the cystic fibrosis. Is its normal function to give people cystic fibrosis? No, it's named that because that's what happens when it's broken. Okay? So that's very common, and unfortunately only the fly people have a sense of humor with their naming. So here's frizzled. Disheveled is DSH, stands for disheveled.
Oh.
Okay? And you know what disheveled means, like sloppy looking, right? Like unkempt. So same thing. So without the growth factor there, disheveled, here you have this complex here of beta-catenin. Here's APC. Here are some of these other proteins in that pathway, this kinase. Beta-catenin gets you phosphorylated, and then it gets ubiquitinated, which we just talked about, and it gets degraded by what? If you get ubiquitinated, you get fed into a...
What's the name?
Find it.
¿De cuál? La cosa que se comía. MD.
No, if you get ubiquitinated, you get fed into that cylinder, chopper thing. That's called a proteasome.
Proteasome, right.
Okay? All right. So yeah, so this is very common for things that we only want to be around for a little while. The proteasome is able to very quickly get rid of proteins that you want to critically regulate, and that's what happens here. So if there's no growth factor, then the beta-catenin gets destroyed. And the target genes are bound to Groucho, right? Like Groucho Marx, of course, right? Again, another fly gene, right? You can always tell. Groucho and these other transcription factors are bound here. The target gene is repressed. If, on the other hand, the signal Wnt is out there, then that causes these to associate, which indirectly, of course, makes this not happen, so you don't get ubiquitination. Beta-catenin gets released, and it goes into the nucleus, and it works as a transcription factor. And this is what drives the cell division forward. This leads to production of MYC and cell cycle progression, and if it's not controlled, cancer. Okay. All right. So remember I showed you a little interaction chart with P53?
Okay.
Here's APC.
Okay.
So again, this would be a good one to put on your final. Right? Again, not to be memorized at all, just a concept map for you, okay? Like P53, this thing is a sensor for multiple different things that are going on in the cell. It's looking at cell-cell binding, metabolism, extracellular matrix, Golgi, cell trafficking, nuclear histones, DNA binding, splicing. I mean, you got it, right? The works. So again, like P53, when this one is gone, through very bad engineering, we're screwed. Okay? And that's why when this protein by itself is defective, you can end up with inherited cancer. Okay. So again, just to take away those things and make it a little bit more digestible, there are multiple cellular processes, migration, transport, gene expression, protein folding, adhesion, whatever, that APC is involved in. And if you look at where the cancer happens, right, where did we say, what kind of cancers did they get? I showed you that disgusting picture with all the polyps.
Oh, the colon.
Colon, right? Okay, colon. So here's a picture of the colon. Okay, so here's a colonic crypt. Okay, here's a colonic crypt. So what these things do, what these crypts do, is secrete mucus and help increase surface area so we can absorb water. Okay, and so what happens is the cell division happens down here. And the stem cells, the precursor cells, right? The epithelium is changing all the time in the gut because it's exposed to all kinds of, it's a terrible environment. For all I know, you guys were at McDonald's before you came to class, right? What have you done to yourselves, right? These cells are dividing all the time. They migrate up. And so you essentially need, this APC is critical here because it is involved in all of the steps that take this cell from down here when it's a baby from a stem cell all the way up to where it's going to eventually be shed. And so these cells are really reliant on all those things. They're modal, they move, they secrete stuff, they're right. And for whatever reason, the cells have essentially put all of their eggs in one basket. Do you know that term?
Mm-hmm. Yes.
Right? It makes sense anyway. Even if you didn't know it, it makes sense. Right? So they put all their eggs in the APC basket. The logic for it, right? No one knows, just like P53, right? This is a guess. We're making a guess. But the guess is that it's a centralized way of responding to stress, right? Because APC is able to sense all this stuff just like P53 senses so many things. So living in the gut is stressful. The cells are exposed to acid, to chemicals, mechanical stress, as we mentioned, right? That constant abrasion. So if you guys eat a bunch of peanut shells or something, then your gut tissue has to deal with that, right? Later. So you know, they're subjected to a lot of stuff. And so the thing is, it may be worth the trade-off, right? It's not clear. It seems to be pretty common now that we're looking at the way these things work, that this is a pretty common system. You do get cancer. The cancer tends to happen in old people, right? After they've had kids. And so the system seems to be worth the trade-off, right? If you get cancer when you're 70, you're no longer reproducing, your kids are old enough to reproduce, we really don't need you from an evolutionary perspective anymore.
Okay, so it would be like the cells being under so much stress that it's better for the system to just, I don't know.
I mean, the thinking is that, like, if you can have, I don't know exactly. I wish I could explain it better, but I don't know. I mean, it's just like a conjecture. People have said, well, you know what? If we have one sensor, then we don't have to try to integrate the signals from 20 different ones and make decisions. We have one major decider. It's like President Bush. He said, I'm the decider, right? So this protein, or P53, seems to be the decider. Mm-hmm. Rather than have one that senses DNA damage and it has to talk to another one and another one, it seems like that evolution has decided to converge those responses onto a few proteins, mm-hmm. which seem to be particularly important. It may be due to the relatament of the development of the tissue, why they use it. It's not clear why different cell types are so dependent on particular genes. Because they get colon cancer, they don't get brain cancer. Mm-hmm. So we don't know. I mean, the answer is we don't know why, but we just see it. Okay? Now, one of the things that was and is a very active area of research is the question of whether or not tumor suppressors can rescue cancer cells in patients. Because that would be a good thing, right? Presumptively, if someone has cancer because they have P53 mutation, we should be able to give it back and they should be better. And when we looked at the fusion experiments, remember we took cancer cell with a normal cell and we fused them, and most of the time they act normal. Mm-hmm. So that would be an indication that if we can get those tumor suppressors in there working, then it's gonna help, right? So So people have looked at it. So does activation of tumor suppressors cause tumors to go away? It's still a very active area of research. Or conversely, before we even get cancer, wouldn't it be smarter to just turn p53 up all the time so you don't get cancer to begin with?
Yeah.
Okay, so that's a good idea. And so people have looked at these things and are continuing to do so. It's not done by any means. People are still looking at this. So they've tried to reactivate p53 using the CRISPR technology. Are you familiar with CRISPR? I think I may have mentioned it last time, maybe, but it's all uppercase, CRISPR. CRISPR. There are videos on YouTube. You need to watch them. It's the future of medicine. Okay? It's a new way of altering gene structure and function. Okay? It's as big, I think it's going to be as big as like PCR when it came out, you know. It's going to be a big deal. Okay? Or already is. So you guys want to know about that so you can look down when other people don't know it and you're like, what's wrong with you? You don't know CRISPR. Right? Okay. So people are trying to turn these genes on, right? So here's just a couple different examples. They're trying to turn on p53 pathway, MASPIN, p16 INK4A, which is, by the way, the other protein that comes out of ARF. It's ARF and INK4A are the two proteins that come out. Remember I said there's two that come out of the same gene?
Yes.
You can forget that immediately, but it's p16.
Oh.
Okay. So here is this is the last story I'm going to tell you today. This is a paper that came out in 2002. It's not perfect, and I know that it's not perfect, the experiments, but it's very interesting. And they asked the question, what would happen if p53 was on all the time? Right? We showed you that it's on only when cells are stressed, right? Is when you get activation of the p53 pathway. But what would happen if it really were on all the time? And so they created animals. These were mice that were transgenic. That means that they have this gene in all of their cells, right? Mm-hmm. Trans means across. Mm-hmm. Genic meaning genome, transgenic, meaning across genomes. So they took this gene from one animal or organism and they put it into the genome of another, transgenic. Okay? And so they have a truncated, truncate means shorten. To truncate something means to shorten it. So they have a truncated or shortened version of p53 called p53M is the name of the allele. The form of the gene is an allele, right? And they have this p53M. p53M is constitutively active. It is always on. Okay? And this is the name of the paper. p53 mutant mice that display early aging associated phenotypes. I think the title sort of gives away a little bit of what happens here.
Yes.
Yeah, they telegraphed that, didn't they? They shouldn't have done that. They should have made it more like, what happens? You know? Yeah. Okay. So, let's look at what happens, because I think this is really instructive. I mean, I think this is a good take-home lesson for you about why humans get cancer to begin with. Okay? Now, here we have p53++ mice. This is survival over time. So this is a death curve. It's called a Kaplan-Meier plot. Right? This means how many mice are alive. Got it? You see it?
Yes.
She's leaning forward. She's trying to see it.
Well, no, I'm only one part, so yeah. But
Okay, but can you get it?
yeah, I didn't think it.
Okay. As long as everybody's awake, I'm happy. Okay. So
We all are.
okay. So here's the wild type p53++, right? Overall, they live the longest. They get about 45% of them get cancer, but that's to be expected, I guess, right? And that's what it is for that strain of mouse. p53 with constitutively active here, this plus M, doesn't live longer. It's actually lives a less time, right? But as you would expect, look at the tumor difference. 45% here, 6% tumors here. So if you have p53 on all the time, you don't get cancer, right?
Right.
Or it's obviously greatly reduced, okay, cancer. But they don't live longer. If you take animals that are heterozygous for p53, this would be sort of like Liezl Romani. You get greater than 80% of them get tumors and they die very quickly. Okay? So what's going on, right? Are you perplexed?
Yeah. Well, a little bit.
Okay.
Okay, you're supposed to be perplexed. Okay? So let's look at these animals. When you look at the animals, this is the wild type animal without any skin, of course, right? I doubt that was voluntary, so I think the animal's dead. This animal has a very different look than this one, okay? This animal has, first of all, the shape of the spine. It's got lordosis, right? It's got the curvature of the spine, or lordosis. It's got this curvature. You see that?
Yes.
This is what happens in old people, right?
Yeah.
Right? It looks old, doesn't it?
Yes.
And look at the subcutaneous fat. Look at this skinny little thing. It has less fat, less muscle. It's like an old person,
Yes.
right? Here's what happens when you look through the animal. You can see the curvature, dramatic curvature. Okay?
Yeah.
And so what's going on? What's going on? Well, it turns out that what we have is, with p53, our normal amount was the best way to go, right? We were best off with what the normal amount that we would have. Or I should say the mice were. Right? But presumptively, this extends to humans. So the idea is that at a cellular level, p53 is responding to oncogene signaling, loss of telomeres, stress, oxidative stress, that is, peroxide, oxygen radicals, etc., radiation, SIR2, which is a protein that's very interesting. You should research, do a Google search on SIR2 and aging. You'll like what you find. So SIR2 is another protein that blocks it. And at the cellular level, it leads to senescence and apoptosis. At the whole organism level, what you get is tumor suppression, but if it's active too much, at least in this scenario, what we saw was accelerated aging. And the reason for that, the reason you see the accelerated aging, let's put yourself inside that mouse. Okay? We're inside that mouse with one of each. Okay? So it's got p53 on. You get a little damage to your DNA. What does your cell do?
It tries to repair it, or?
No, you don't. You have p53 on all the time, you M allele U.
Oh, isn't it that?
Right? So if p53 was constitutively active, essentially it has a hair trigger to cause these cells to undergo senescence or apoptosis. That does block tumor suppression. But essentially, and remember in the very beginning of class, in the very beginning, I talked about that Hayflick limit, that cells only can divide so many times. Yeah. Right? See, don't you love the way I tied that together? Huh? Beautiful. Seamless integration, right? So we have this limit, right? So if you have p53, if the trigger is too easy to pull that apoptosis trigger, right? Or senescence, that cell's out of the division loop. The cells, the organs, burn through their divisions too fast, and you essentially lose the ability to replace tissues that get damaged. When things go bad, you no longer have the rejuvenation capacity. And that seems to be what happens in those animals. The story is a little more complicated than I presented it because that M allele is weird and does other things. But as a take-home message, I think it's very clear that the longevity of the animals is not based on our preference of, wow, I'd like to live 30 more years. I'm 52. I'll be 53 on Saturday, by the way. My birthday is in a couple days. So yeah, thank you. So I'll be. You can sing Feliz Cumpleaños I would love to live 30 more years, but evolution really doesn't give a crap about that. It wants me to be healthiest when I'm reproducing and when I'm raising my offspring to reproductive age.
Mm-hmm.
Right? And so that seems to occur. They seem to live longer, be better with the sort of the default pathway, the default amount. Right? If you turn it down, you're going to get tumors and you're going to die too young. If you turn it up, you're going to die because you're going to accelerate aging. And so, like I said in the title here, life is sort of about balance. And I think that's probably a good place to stop. I don't actually